The assessment of antibacterial activity is carried out through bacterial reduction rate analysis. This method evaluates how effectively an antibacterial substance can reduce or eliminate bacterial cells from a given suspension. When exposed to antibacterial agents, bacterial cells can be damaged or destroyed, resulting in a measurable reduction in their population. Welcome to Microchem's experiments. Be with us, be a laboratory expert. This analysis follows a structured 6-step process. The steps are media and diluent preparation, bacterial culture preparation, bacterial culture dilution, control test, sample test, and result interpretation. To conduct the test accurately, we need biological safety cabinet, colony counter, balance machine, autoclave, Bunsen burner, inoculating loop, micropipette and micropipette tips, petri dish, glass rod, bacterial reference culture, tryptone soya agar, tryptone soya broth, and sodium chloride. Let's begin by preparing the culture media and diluent. We need tryptone soya broth for preparing the bacterial reference cultures. First things first, always follow the manufacturer's instructions when working with commercial dehydrated culture media. Now, measure out 1.5 grams of dehydrated tryptone soya broth powder. Next, pour 50 milliliters of distilled water into a clean glass bottle. Add the powder to the water and mix gently. To fully dissolve the medium, heat the mixture slightly. You can use a microwave, a water bath, or a hot plate. Don't overheat, just warm it enough to dissolve everything completely. Once dissolved, dispense 10 milliliters of the broth into each screw cap test tube. Make sure the caps are placed loosely. This allows steam to escape during sterilization. Now it's time to sterilize. Place the tubes in an autoclave and set it to 121 degrees Celsius at 15 psi for 15 minutes. Once sterilization is complete, allow the tubes to cool down. Then, tighten the caps and store the media at 4 degrees Celsius until you're ready to use it. Let's begin preparing tryptone soya agar, or TSA. Start by dissolving 40 grams of dehydrated TSA powder into 1 liter of distilled water. Stir well and gently heat the mixture until the medium is fully dissolved. Next, sterilize the solution by autoclaving it at 121 degrees Celsius and 15 psi for 15 minutes. Once autoclaving is complete, carefully pour the hot medium into sterile petri dishes. Allow it to cool and solidify at room temperature. Finally, incubate the plates for 24 hours to check for any signs of contamination before use. Always make sure to follow the manufacturer's instructions when using commercial dehydrated culture media. For a step-by-step guide on preparing agar plate culture media, be sure to check out our dedicated video on the topic. Video link is given in the description section. Next, we'll prepare a normal saline solution to use as a diluent for this test. Start by dissolving 4.25 grams of sodium chloride in 500 milliliters of distilled water. Dispense 9 milliliters of the solution into each Falcon tube. Then, sterilize the tubes by autoclaving at 121 degrees Celsius and 15 psi for 15 minutes. After sterilization, allow the tubes to cool completely. In this step, we'll prepare a bacterial culture to test the antibacterial properties of our sample. Start by sterilizing an inoculating loop. Burn an inoculating loop to red hot to sterilize. Make sure to cool it down before use by holding it inside a biological safety cabinet. Here we have a reference strain of E. coli. This will be the test organism for our antimicrobial sample. Pick a single colony from the agar plate using the cooled loop and aseptically inoculate it into a sterile nutrient broth. Now, incubate the culture at 37 degrees Celsius for 24 hours. This is the optimal temperature for E. coli growth. After incubation, bring the culture back into the biosafety cabinet. You'll notice the broth is turbid. This cloudiness confirms that the bacteria have grown. At this point, the bacterial concentration is roughly 1 × 10⁸ CFU per milliliter. Next, we need to dilute the bacterial suspension to a lower concentration for testing. Start by vortexing the culture tube to ensure a homogeneous mix. Then, perform a serial dilution to bring the concentration down to about 1 × 10⁵ CFU per milliliter. If you're not familiar with the serial dilution technique, we've linked a detailed video in the description below. Be sure to check it out. This diluted suspension is our working concentration, and it's what we'll use to test the antimicrobial sample. Now let's set up a control test. This will follow the exact same steps as the sample test, just without the antimicrobial compound. Take your working E. coli suspension and dilute it further to 1 × 10 cubed CFU per milliliter, or 1000 CFU per milliliter. From this, spread 0.1 milliliters onto a tryptone soya agar plate using a sterile glass spreader. This is your trial 1 plate for the control. Repeat this for a second plate, trial 2. This gives you a reliable baseline to compare against. More on the spread plate technique is also available in the description video link. Now it's time for the actual sample test. To your working bacterial suspension, add 0.5 milliliters of the antimicrobial sample. Vortex the mixture thoroughly and let it stand for 3 hours. This is the contact time, during which the sample interacts with the bacteria. Depending on the antimicrobial's mode of action, you might choose to extend this contact time. Longer durations can sometimes give more noticeable effects. After 3 hours, vortex the suspension again. Dilute it to 1 × 10 cubed CFU per milliliter, just like we did for the control. Vortex the mixture thoroughly. Then, perform a serial dilution to bring the concentration down. If you're not familiar with the serial dilution technique, we've linked a detailed video in the description below. Be sure to check it out. Now, spread 0.1 milliliters of this diluted suspension onto a fresh tryptone soya agar plate using a sterile glass spreader. This is your trial 1 plate for the sample. Repeat this for trial 2. With both control and sample plates ready, incubate all plates at 37 degrees Celsius for 24 hours, the optimal temperature for E. coli growth. Once incubation is complete, carefully take the plates out and bring them back into the biological safety cabinet for observation. Now, you're ready to compare bacterial growth between the control and sample plates to evaluate the effectiveness of your antimicrobial substance. Here, we have the trials 1 plate of the control test. The bacterial colonies on this plate appear quite dense. To improve visibility and make colony counting easier, it would be better to dilute both the sample and control solutions by an additional 10-fold. And this is the trial 2 plate of the control test. As you can see, both trials show a similar number of colonies, indicating the consistency and reliability of our test results. Now, this is the trials 1 plate of the sample test. Compared to the control, we observe a significantly lower number of colonies. This suggests that the tested sample possesses strong antimicrobial activity against E. coli, effectively inhibiting most of the bacterial growth. Only a few bacteria were able to survive and form colonies. This is the trials 2 plate of the sample test. On this plate, we can observe a few large colonies that appear atypical compared to our tested E. coli colonies. Therefore, we should exclude these foreign colonies from our count to ensure accurate results. These unusual colonies may have originated from contamination sources. As you can see, both trials show a similar number of colonies, indicating the consistency and reliability of our test results. Let's begin counting the bacterial colonies in the control test. On the trial 1 plate, we counted 864 E. coli colonies. Now moving on to the trial 2 plate of the control test. We found 921 colonies in trial 2 of the control test. Next, we'll count the colonies in the sample test. On the trial's 1 plate of the sample test, we observed 36 colony-forming units. On the trial's 2 plate, we counted 42 E. coli colonies. Now we're ready to calculate the bacterial reduction rate. After counting, the average number of colonies in the control test is 892, while the average in the sample test is 39. Using the formula shown on the screen, the bacterial reduction rate of our tested sample against E. coli is calculated to be 95.62%.